For video 4, I chose the topic of receptors, including soluble, gated ion channels, RTKs, and G-protein coupled receptors. So signaling pathways in cells begin with the binding of a ligand to a receptor. The binding of ligands to receptors changes the shape and activity, allowing it to transmit a signal or produce a change in the cell. There are two main types of receptors: intracellular, which are in the cytoplasm and the nucleus, and cell surface, which is in the plasma membrane, including RTKs, G-protein coupled receptors, and gated ion channels. Gated ion channels open in response to the binding of a ligand. It has a membrane with a hydrophilic channel in the middle, and the channel lets ions cross without having to touch the hydrophobic core of the phospholipid bilayer. When a ligand binds to the extracellular region of a channel, the structure changes so ions such as calcium or chloride can pass through. Next is G-protein coupled receptors. These transmit signals in cells through proteins called G-protein. It binds to GTP, which can hydrolyze to form GDP. When the G-protein is attached to GTP, it's active, and when it's attached to GDP, it's inactive. And when the ligand is not present, the G-protein coupled receptors wait at the plasma membrane in an inactive state. And when the ligand is present, GDP is exchanged for GTP. The now active G-protein separates into two pieces, a subunit and another with beta subunits, which are then freed. And these freed subunits interact with other proteins, signaling pathways that lead to a response. And then eventually the subunits will hydrolyze GTP back to GDP, which will make the G-protein inactive. And it's a cycle that repeats itself over and over in response to ligand binding. Next is RTKs. They're a class of enzyme-linked receptors. It stands for receptor tyrosine kinase, and kinase is a name for an enzyme that moves phosphate groups to proteins or other targets, and RTKs specifically transfer phosphate groups to the amino acid tyrosine. The process includes signaling molecules binding to extracellular domains of nearby receptor tyrosine kinase. Two neighboring receptors then come attach phosphates to tyrosines in each other's intracellular domains, and the phosphorylated tyrosine can now transmit signals to other molecules in cells. This whole process plays a huge role in growth factor signals, so overgrowth is typically associated with multiple types of cancers.